अस्सलाम वालेकुम, क्या हाल चाल है? ठीक ठाक है. So the topic of our discussion today is taken from the FSC part 2, and the chapter is third. And if we really follow the sequence of the FSC, it probably the chapter number 17, which is the coordination and control. And from coordination and control, we will discuss today the nerve impulse. Actually, first of all, I would like to tell the basic of the chapter. Actually, this chapter is the basics of the neurology. As far as you are going to the higher classes, you will learn the neurology in detail. So this is the very basic things, like you will learn the neuron structure, and the neuronal physiology, or later on you will discuss the nervous system and endocrine system, and then you will end up with the behavioral biology. So out of this chapter, we will discuss the topic nerve impulse. So the topic which we are going to discuss is about nerve impulse. Nerve impulse. This is page number from I think 62 to 65 we will cover today. Nerve impulse is also known as action potential. Action potential. This is probably the topic that you guys don't know about anything, because there is nothing about this topic in the matriculation. So the FSC students, I don't think so, they know about the nerve impulse. Before really going detail about the nerve impulse, okay, let me tell you what is nerve impulse. Nerve impulse, if someone asks you what is nerve impulse, it is simply the messages. Like if someone touches me here, and I'm saying no one touches me here, but if someone touches me here, so the message goes from all the neuronal pathway to the higher central nervous system. So this, what is this traveling? This is the nerve impulse, which goes through from this nerve ending tip or the finger tip to the higher centers of the brains. So nerve impulse, simply it is the action potential. It is the potential difference or the potential across the cell membrane that travels, that in action. So this is simply nerve impulse. But before discussing the nerve impulse, everyone should know the potential difference. Actually, we will start with the very basics. So don't make it very hard, but try to learn this. I am just taking the example of one cell. If just this is your cell. Again, I am telling you, we are not going to discuss about the nerve impulse right now. Before this nerve impulse, we must learn or understand the one terminology, and that is the resting membrane potential, or we can say membrane potential. Membrane potential, or because this membrane potential present almost in all cells when the cells at resting. So the most probably we call it resting membrane potential. So before nerve impulse or action potential, you must know about the RMP, that is resting membrane potential. I am taking the cell, this is any cell of the body. Now one more thing, nerve impulse, resting membrane potential, this is we discuss at the general level. Actually, whenever you discuss at the vertebrate level, all the vertebrates, the action potential, the nerve impulse and the RMP is the same, with little exceptions, because even in the human bodies, different cells show the different action potentials. But the general mechanism we discuss now at the FSC level and very little bit detail at the OA levels. First of all, taking a cell, this is any cell, this can be the any cell, just suppose, and this is the neuronal, no, this is a skin cell, just suppose, or this is the muscle cell. Okay, I will taking the muscle cells. Now, you know the muscle cell, and there is a nucleus, and this is what, this is your genetic material, and in the genetic material we know there is a genes present, and just suppose this is a particular gene. Whenever this gene expresses, you know there is a central dogma, gene expresses in the form of messenger RNA, and then this messenger RNA comes in the cytoplasm and the protein is formed. This is transcription and translation generally. Now this protein will embedded or implanted into the cell membrane, into the cell membrane of the cell. Now this protein is present almost in all the cells. Even my trillions of the cell has this pump, and all vertebrates. This is a very, you can say, conserved protein. This is the protein which is embedded in the cell membrane, and the many amounts, the thousands and hundreds amount of this enzymes. I will tell the name later, but right now you just concentrate the enzyme will express as the gene is expressed in the mRNA and the protein, and this protein is embedded in the cell membrane. Now, what does this protein do? Actually, this protein throws sodium out and potassium in by using ATP, by using the ATP. Now actually this is ATP and converted into the ADP plus inorganic phosphate. Now what does this pump do? This pump utilize the energy. No, one more thing. Actually you must be knowing about the pump, the terminology pump and channel. Pumps and channels are present in the plasma membrane. Like just suppose this is a pump and this is the channel. What is the main difference? There is a difference, and you must be knowing pump. Whenever we call it the pump, this is a pump. This is particular pump, particular ion pump. What does it mean? That its activity is such like that it throws the anything, if ions, if hormones, if anything, it throws a particular thing against the concentration gradient. What does it mean? Like just suppose this star concentration is high in the outside and this star concentration is less inside. So this pump, usually normal star will tend to move inside the cell because star concentration is high outside the cell and star concentration inside the cell is low. So usually the star will tend to come in, but this pump still, despite the fact that the star is high concentration, high amount in outside, it will throw this star outside, and this against the concentration gradient we use the energy. So pump is always activating as pumping the things against the concentration gradient. But the channels is simply, if the star is concentration high and it will tend to move inside. So this is the channel. Channels will move things along the concentration gradient. So this is the difference. Now this pump, actually this enzyme named as sodium potassium pump. This is the sodium potassium pump, and of course this is using the ATP. We call it sodium potassium ATPases. ATPases. Now remember, this enzyme throws the sodium out and brings the potassium in. Again, this pump is unfair as the people's are. What is the unfairness of this pump? Actually, it throws 3 sodium out and bring 2 potassium in. There is unfairness of this pump. Actually, whenever this pump switches or conformational changes or turning up, it will bring the 3 sodium out and brought the 2 potassium in. What does it mean? Actually, you must be knowing at the normal state, normal healthy physiological person, your all cells, your all cytoplasm is neutralized. Electroneutrality is present in all of your body. There is no, we can say there is no imbalancing of the ions or electronegativity. But this pump will produce actually 3 sodium out, 2 potassium in. It means the cell loses more cations and gaining less cations. It means the cell will become more electronegative. Cell will become internally electronegative. Because this is very simple, if this pump works 3000 times, it will throw 3000 sodium out and gaining the 2000 potassium in. It means net gain, there is net loss of the positive ions, net loss of the cations. So internal environment will become electronegative. So how much electronegativity is produced by this pump, this nasty pump? Actually only minus 5 millivolt. Minus 5 millivolt is produced by this pump. This is electronegative. This is very little contribution of this pump. Again, let me repeat it. Sodium potassium pump, what it does, it throw the 3 sodium out, bring the 2 potassium in, and whenever it brought these things, it utilized the ATP, and as the result, the net gain or loss, we will decide this is a net loss. Actually, whenever this pump works, it throw the more cations out and gain the less cations in. So we can say the cell membrane or the plasma membrane internally is negative charged, electronegative, and the contributing factors are somehow basically there is the different anions and proteins and phosphates are present behind this. So the sodium out and these negative ions also contribute this electronegativity, and you must be knowing the electronegativity is produced, or genesis of the electronegativity by this pump is only minus 5 millivolt. This is not the resting membrane potential. We are discussing the resting membrane potential, but this is not the resting membrane potential. This is the contribution of this. Now what is the situation of this cell? Actually you must be knowing, okay, one more thing, Before this, I would like to tell that the normally cell inside has more potassium and outside has less potassium. And outside there is a high sodium concentration and inside there is a less sodium concentration. Despite this fact, this sodium potassium ATPases or enzymes work as a pump and throw the sodium out, bring the potassium in, uses the ATP and bring the electronegativity inside the cell membrane about -5 millivolt, right. Now, another enzyme will, you can say, activated, and of course messenger RNA, and this will make the protein, and these are the channels, embedded in the cell membrane. Now, what are these channels? These channels are the potassium. Actually, you must be knowing there are channels, different channels present. I am not going into detail. There is a leaky channels. Leaky channels is all the time open. Then we have the chemical operated channels. Chemical operated channels mean these channels activity or these channels opening and closure is depending upon the chemicals. It may be the neurotransmitter, it may be the hormone, it may be any toxin, drug. If this is the present, then the channel is open and closed depending on the chemical nature. And then there is the voltage gated channels. Then there is voltage gated channels. Voltage gated channels mean these channels are sensitive to the voltage, the potential across the plasma membrane. So, we have ultimately these three kind of channels. Now, what are these channels? These are the potassium leaky channels. These are the potassium leaky channels. Remember one thing, that when a cell is at resting state, all the cells are permeable to potassium. This is the rule. I am summarized these events right here. First of all, we talk about the sodium potassium pump. Don't confuse, we are not discussing nerve impulse right now. We are discussing right now RMP, resting membrane potential. The events of the resting membrane potential, the first level we have the sodium potassium pump, which simply throws sodium out, 3 sodium out and bring 2 potassium in by using the ATP simply. Second point, you must be remember that at resting state, kiski resting state, cell. At the resting state of the cell is always permeable to potassium. A little sodium will come and out, but this is negligible. This is not the considerable point. The main thing, the resting state, the cell is always permeable to potassium. It's mean. Now, first of all, we will talk about the potassium. Let me make the diagram more clear. Just suppose this is a cell, and first of all, what we have seen this pump. This pump throws sodium out and potassium in simply, and using the ATP. Now, we have potassium inside and sodium outside. Okay, and these are the potassium leaky channels. There is no doubt the sodium mechanical channels, voltage gated channels and sodium related channels are also present. But at resting state, those channels are closed, those channels are not operated. At resting state, only the potassium leaky channels are operated. Even though potassium leaky channels are present, potassium voltage gated channels are present. Still at the resting state, potassium voltage gated channels are closed and the potassium leaky channels are open. Now concentrate. You know, potassium concentration is high inside. Due to this genesis, this pump, there is only -5 millivolt. So, this is electronegative inside and electropositive outside. Now, there is a 2 gradient. Actually, concentration gradient of the potassium force potassium to move outside, because the concentration of the potassium inside is high and everything moves from high concentration to the low concentration. Beside this, the concentration gradient, there is a little electrical gradient also present that attracts potassium inside. What is electrical gradient? Because inside cell membrane is negative, and negative attracts the positive, so there is a little electrical gradient also act on the potassium, that attracts the potassium or holds the potassium inside the cell, that it not escape the potassium easily. But both of them, this is a concentration gradient and this is the electrical gradient. Out of these, concentration gradient is strong, because potassium concentration inside the cell is so much high that it tend to move outside, because the electrical gradient is not so high. So what happens when, as far as potassium moves out, cell membrane will become more and more electronegative. First of all, it will -5, then might be there is -10, minus, just suppose -40, -50, -60, -70, -90. At a state will reach that the concentration gradient, now potassium is trapped inside the channel, and the concentration gradient will exactly equal to the electrical gradient. There is no net movement of the potassium. The net movement and the resultant forces will become zero. That potassium will not move inside, neither move outside. It will trapped. So what we achieved? We achieved the equilibrium potential. This is called the equilibrium potential. This potential is called diffusion potential of the potassium. Remember, this is the diffusional potential of potassium. And as far as potassium moves out, cell membrane will become more and more electronegative, and that state will reach that the concentration gradient will exactly equal to the electrical gradient, and potassium movement will stop. Ionic movement will stop, it will become zero. That particular point is known as, this is the equilibrium potential for potassium, and it also known as Nernst potential. Is also known as Nernst potential, equilibrium potential of the potassium. This is almost ranging from -70 to -90 millivolt. The equilibrium potential of the potassium is also the resting membrane potential. So at the resting state, the cell membrane is not permeable to sodium. These 3 points are very important. Not permeable to sodium. Resting membrane is not permeable to sodium. Resting state cell is always permeable to potassium and sodium potassium pump. Now, do you think who is the responsible for genesis of this particular resting membrane potential? Look, there is a 2 factors. We have discussed one at this point. This is the sodium potassium pump, and at this point potassium level. Now again listen, I will repeat it because it is worth repeating. Beside you are not understanding this one, you cannot understand the nerve impulse or action potential. First of all, you will clear cut concept about the resting membrane potential. Resting membrane potential, first of all, we have the sodium potassium pump. What sodium potassium pump do? It throws 3 sodium out, bring 2 potassium in, and uses the ATP, using the ATP as the energy source, and creating, like due to this activity, more cations loses and less cation gains. So the cell membrane internally electronegative. How much it is? About very little contribution about the sodium potassium pump is about -5 millivolt. But potassium inside is the high concentration. Again, the on potassium concentration gradient is high, but electrical gradient also works because the cell internally electronegative. But the electronegative is not sufficient that it holds the potassium inside. Potassium will move outside. So potassium will escaping the outside, bringing the cell membrane more and more electronegative, and the state will reach that the potassium concentration gradient will exactly equal to the potassium electrical gradient, and the potassium movement will stop, and this is called the equilibrium potential. And remember, resting membrane potential is always near to the equilibrium potential for the potassium, and it is almost ranging from cell to cell, -70 to -90 millivolts. Now, if someone asks you who is the prime contributor for the genesis of RMP, is it sodium potassium pump or the potassium diffusional potential? It is the potassium leaky channels. The direct role in the RMP genesis, direct role in the resting membrane potential genesis is the potassium diffusion potential and the leaky channels. If potassium will not escape, just pause, if potassium will not escape, these channels are closed and already at the resting state, other channels are also off. If these channels are also off, do you think -5 reaches to -90? Not at all, because potassium remain inside, cell will become not so much polarized. So this state, this is actually negative inside and positive inside, and there is a potential difference across the cell membrane. That's why we call it potential difference, or we can say the membrane potential, because this is the resting state when the cell is not excited. Remember, almost every cell has the resting membrane potential. It means that all the cells do these things, but only the excitable cells are only two. Throughout the kingdom Animalia, only two cells are excitable. One is the neuron, one is the muscle cell. What does it mean, excitable cell? Excitable cells, that it is responsive to stimulus. Like if there is a particular stimulus, it responds. Like this resting membrane potential will convert into the action potential. We will see later how. Just right now, you just remember that this is the potential difference across the cell membrane at resting state. What is the cell condition? Cell will internally more polarized. This is the polarity achieved inside the cell, polarized. Remember, at the resting state, during the resting state, cell internally what? Negative. Internally what? Internally externally it is polarized. Who is main responsible for this genesis? This is only potassium diffusional potential. Potassium diffusional potential, leaky channels. Now what is the role of sodium potassium pump? Actually it is a very innocent pump, and it has the indirect role, very influential role. Why? You know, potassium inside cell is very high. Listen, Masha, आप भी इसको जरा गौर से सुनना. You know, potassium inside cell is very high concentration and outside is very low concentration. And diffusion of the potassium will bring the membrane -5 to -90. But who brought this potassium in? Sodium potassium pump. Every time it rotates, it brings potassium in. Every time it rotates, bring potassium in. So the concentration of the potassium goes high, and potassium will move outside. So the indirect role of the sodium potassium pump is to brought the potassium in to raise the concentration inside, and then the potassium diffusional potential brings the main RMP. The role of the a sodium potassium pump is almost only -5 millivolt, but up to the -7 to the -90 only achieved by the diffusional potential of the potassium.